Hello students, we'll be starting with the first topic of pharmacology 4, that is principles of chemotherapy. The learning objectives include to know the history of chemotherapy. Second, to learn some terms and concepts associated with chemotherapy. Third, to study the modes of action of anti-infective agents. Fourth, to classify various antimicrobial drugs. Fifth, to study the bacterial sensitivity testing. Sixth, to study the bacterial resistance to antimicrobial drugs through its mechanism of resistance and ways of prevention of resistance. The history of chemotherapy. It was a famous German chemist Paul Ehrlich from 1854 to 1915 period worked on many anti-infective drug treatment of systemic infections. Before his work would be recognized, there were many antiseptics and disinfectants into the picture for the treatment of infectious diseases, but they were too toxic for internal administration for systemic infections. Later on, Paul Ehrlich he came up with a compendium called as Therapia Sterilisans Magna, which offered a challenge to chemists for the control of infections with a single dose of a non-toxic chemical, which he called as a magic bullet. He also synthesized hundreds of chemicals and tested them on laboratory animals, which were injected with pathogenic organisms. And later on, there were many other scientists who also employed Ehrlich's concepts and methods during the next quarter of a century and went on to develop drugs for the treatment of malaria, amoebiasis, and other protozoal diseases. Finally, in 1935, Gerard Dumac from Germany again, he came up with a red azo dye called Prontosil, which was capable of protecting mice against streptococcal infections. And that was a period when sulfanilamide was also introduced. And later came the antibiotic penicillin in 1941. So basically, the concept or the principle of chemotherapy it moves around a term called as selective toxicity. Now, selective toxicity is the term which has been employed to prepare synthetic compounds that will selectively act on parasitic cells without injuring the host cells. And this concept was the theme behind the chemotherapeutic principles. There is one more term called as chemotherapeutic index. Ehrlich believed that the anti-infective substances which were produced should have greater effect on the parasites and have minimal effect on the host cell tissues. That is, it should be maximally parasitotropic and minimally organotropic. So therefore, he devised a index called as chemotherapeutic index, which was the ratio of maximum tolerated dose with minimum curative dose. In the sense that the anti-infective substance which was produced would be sufficient enough or lethal enough to kill 0.1% of the host cells and show its therapeutic capability in 99% of the bacterial infective cells. So this index gives a better idea also on the margin of safety of the compound. Coming to the terms and concept of principles of chemotherapy, the first term used is chemotherapy. It basically implies the use of natural and synthetic chemicals which interferes with the functioning of foreign cell population. Originally, according to Eric, this term was applied only to the use of synthetic chemicals which are employed in the treatment of infectious diseases. But current usage, it makes no distinction between chemotherapeutic agents of synthetic origin and the antibiotic. Second is antibiotic. Basically, these are chemical compounds produced by living microorganisms, which could be either bacteria, fungi, or actinomycetes, which at high dilutions, look at this, which at high dilutions are capable of inhibiting or killing bacteria and other microorganisms. Next is synthetic anti-infective agents. These are drugs which differ from naturally derived antibiotics only in their origin, and they are synthetic in nature. And they differ from naturally derived antibiotics. The term antimicrobial agents includes both types and covers all antibacterial, antifungal, antiprotozoal, and antiviral drugs. Next, selective toxicity. We have discussed about this. It refers to the ability of the chemicals to selectively strike at foreign cells in a way to harm them, kill them, without causing any significant damage to the host cells. Next is potency. This is basically, it's an activity per milligram. It is expressed in activity per milligram and it is expressed on the basis of minimum inhibitory concentration at which it is capable of inhibiting the multiplication of microorganisms. The term bacteriostatic and bactericidal activity, it refers to how the microorganism is either inhibited or it is killed. Static refers to the ability of the compound to inhibit the growth and multiplication of microorganisms, and cidal means actual killing effect on the microorganism. Next term, antibacterial spectrum. It refers to the range of activity of a compound. Like it could be a broad or a narrow spectrum activity. For example, penicillin G has a narrow spectrum. So because it primarily affects only gram-positive organisms, whereas tetracyclines are broad spectrum antibiotics since they act not only on gram-positive but also gram-negative bacteria. The next part, modes of action of anti-infective agents. Most of the commonly used antibacterial agents act by one of these following mechanisms. One is inhibition of bacterial cell wall synthesis. Second, inhibition of cytoplasmic membrane function. Third, inhibition of nucleic acid synthesis. Fourth, inhibition of protein synthesis. Fifth, control of microbial enzyme. And fifth, substrate competition with essential metabolite. The first one, inhibition of bacterial cell wall synthesis. In this, we are aware that bacterial cell walls differ from mammalian hosts in their content called as, which is an essential component, which is called as a peptidoglycan. The cell wall is made up of peptidoglycan, and one of the essential component of peptidoglycan is murin. This gives additional strength and acts as a skeleton basically to protect the cytoplasmic membrane. So inhibition of bacterial cell wall synthesis or injury to the cell wall by the anti-infective agents diminishes this protection. And without the protection, in a relatively hypotonic environment of the body, these bacterial cells will explode. So this is basically the mechanism, because we are aware that the inside osmotic pressure of the cell is roughly around 3 to 5 times greater than the outside environmental osmotic pressure. So, and that differs in a gram positive as well as gram negative bacteria. And one of the important functions which taken care by the cell wall is maintaining this osmotic pressure as well. So if the cell wall gets weakened, so naturally there will be an outburst and the contents will come out and the cell will die. So some of the antibiotics which interfere with the cell wall synthesis include penicillins, cephalosporins, cycloserine, pacitocin, and vancomycin. Second, inhibition of cytoplasmic membrane function. In this, the cytoplasmic membrane, which is placed below the cell wall, encloses the cell, and this functions basically as a very selective permeable membrane and selectively allows the entry and exit of essential moieties. Now, the bacterial cell membrane is a semi-permeable triple layered or a lipoprotein structure which is located between the cytoplasm and the cell wall, and it also poses as an osmotic barrier between the interior and the exterior of the cell. So any disorganization of this membrane leads to disorganization of its function, and the membrane function, it fails in its way, and the contents of the inside of the cell are coming out in a similar way as we had seen it earlier. So drugs which have such an action include polymyxins, colistin, or drugs like novobiosine. One important thing to note in this is drugs like amphotericin B and nystatin. They specifically will bind to certain sterols, and these sterols are present in yeast and fungi, and but not they will not be effective against bacteria because bacteria do not have any sterols in their cytoplasmic membranes. So they are better put as antifungal agents, amphotericin B and nystatin. Third mechanism of action is inhibition of nucleic acid synthesis. We are aware about how the nucleic acid plays a very important role in multiplication and the survival processes of the bacteria. Replication of DNA takes place. What happens basically is now the process in which the nucleic acid synthesis occurs is in a way inhibited by these antibiotics. So several antimicrobial agents and anti-cancer drugs interfere with the nucleic acid synthesis. Now, for example, drugs like nalidixic acid and various anti-cancer agents, they inhibit the nucleic acid synthesis in one way or the other. I'm not going into the depths of uh, places where the nucleic acid synthesis is inhibited. The anti-cancer drugs, they will have a very low therapeutic index, and therefore they are not suitable at all for the treatment of most of the infections. Therefore, they are not ideally used as an anti-infective drug because of their low therapeutic index property. So they are not classified under this anti-infective drugs. Next class is inhibition of protein synthesis. Now, synthesis of bacterial protein occurs on the ribosomes by interaction of transfer RNA, messenger RNA, ribosomal RNA, and all. We are aware of the functions of individual RNAs, messenger RNA formation on the DNA, or transfer RNA, which is responsible for carrying out the appropriate amino acid to the ribosomes, or ribosomal function, ribosomal RNA, which has two subunits, 30S subunit and 50S subunit, and it holds the transfer RNA and the mRNA and assists in the process of protein synthesis. Most of the aminoglycosides, antibiotics under antibiotics is a class called aminoglycosides like streptomycin, gentamicin. They inhibit the protein synthesis function. For example, streptomycin, gentamicin, they will bind to the 30S subunit. Chloramphenicol will bind to the 50S subunit and interfere with the function of these ribosomes. So that is how they inhibit the protein synthesis. Next is control of microbial enzymes. What happens is now there are certain metabolites which are used up by the bacterial cells for their normal metabolic processes, and they are synthesized within their body cells, within their cells. So the formation of these important moieties within the bacterial cells is controlled by certain factors or cofactors for which folinic acid is essential. And the formation of folinic acid from folic acid is in turn controlled by enzyme activity. Like, for example, you have a substrate called as PABA, which is responsible for the formation of folinic acid. Now, in the transformation of folic acid to in the transformation of process of PABA to folinic acid, there are several steps, two steps, and in two enzymes they act in these two steps. So these enzymes are inhibited by certain antibacterial agents. So these microbial enzymes, what is meant here is the microbial enzymes are under the control of the anti-infective agents. For example, a drug called trimethoprim controls or inhibits a particular enzyme which helps in the conversion of folic acid to folinic acid. Next is substrate competition with essential metabolite. Here, in this case, the sulfonamides class of drug compete with a natural substrate called as PABA, which is an essential metabolite for the synthesis of folic acid in certain bacteria. The sulfonamides prevent this synthesis by substrate competition. And the bacteriostatic action of sulfonamides can be reversed by the excess of PABA also. A point to be noted. So in this, you can see the diagram very well. It's a diagram of a bacterial cell, a prokaryotic cell having a cell membrane, well-defined cell membrane and a cell wall, wherein the drugs can interact or act on these areas of cell wall. The plasma membrane can interfere with the process of protein synthesis or inhibit the nucleic acid replication and transcription process or inhibit the synthesis of essential metabolites. One more similar diagram which tells us about the mode of action of anti-infective agents. Classification of antibiotics. We have several types of classification, one based on the type of organisms against which it is active, like it could be acting against either gram-positive or gram-negative, or either against cocci or either against actinomycetes. Based on the mechanism of action, we have already seen it in the earlier slides. There are 4 to 5 different uh, uh, um, areas within a cell where the antibiotics can act either on the cell wall or cell membrane or on the nucleic acid or on the protein synthesis part, or based on their type of action, whether it is a bactericidal effect or a bacteriostatic effect. Also classified based on its source. Primarily, there are 3 types of microorganisms where antibiotics are isolated: antibiotics from fungi, antibiotics from actinomycetes, and antibiotics from bacteria. Based on the spectrum, like either it is classified as a broad spectrum antibiotic or a narrow spectrum of antibiotic against which it is effective against the microorganisms. A broad spectrum means it will have a range of microorganisms against which it is based on the activity, whether it is an anti-cancer or an antifungal antibiotic or an. Now, this sensitivity testing in vitro is the most certain way to select the right drug against an organism which would be responsible for an infection. However, most of the time such tests are not always necessary, and the doctor often prescribes us the drug just by experience. Now, for sensitivity and resistance studies, 2 microbiological methods are used. They are called as the Kirby-Bohr technique, which is a standard test for determining the infecting organism's susceptibility to drugs. One method is the uh, paper disc method, wherein the drugs are impregnated on paper discs and they are placed on the surface of the agar plate that has been streaked or swabbed with bacteria obtained from the patient. The plate is then incubated overnight and read after 12 to 18 hours. Now, during this time, the bacterial colonies grow and except where they come in contact with a growing inhibiting drug. Thus, drugs which are effective against a pathogen produce a clear zone of inhibition around the disc, whereas organisms which are resistant, they tend to grow along the side of the disc. In the second method, the isolated organism is inoculated into tubes or into plates of the culture medium containing sequentially diminishing drug concentrations. So, following incubation, the minimum inhibitory concentration of the drug is determined. One specific thing to be noted is there are two specific conditions in which routine sensitivity testing may lead to misleading results. One is the subacute bacterial endocarditis, and the second condition, the urinary tract infection. Subacute bacterial endocarditis has been cured by bacteriostatic drugs very rarely. Now, in urinary tract infections, antibacterial agents are excreted in the urine, and higher urinary concentrations are attained in the blood. So, what happens is now these antibiotics, like for example streptomycin, they possess greater antibacterial activity at an alkaline pH, and they are done, their activity is tested in vitro at a pH of 7.2 to 7.4, whereas the urinary pH is around 5 to 6. So it is most likely that such type of drugs like streptomycin will be effective in vitro than in vivo, unless and until the urine is simultaneously alkalinized. On the other hand, agents like tetracycline or nalidixic acid, which are more effective in acidic pH, may appear to be ineffective when studied in vitro, whereas in acidic urine in vivo they might be effective. Now, coming to bacterial resistance to antimicrobial drugs. Bacteria are susceptible to some antibacterial drugs and others are resistant. What I mean to say is bacteria are susceptible to some antibacterial drugs and resistant to others. But strains may develop that are resistant to drugs which are supposed to be normally effective against that species. Now, bacterial resistance is a major medical problem, and this has hindered the usefulness of many antibiotics. There is another term called as cross resistance. This is a term which is applied to the phenomenon in which bacteria resistant to one drug are found to be resistant to the second drug without having been exposed to the later. So, cross resistance may develop between chemically unrelated drugs also, like erythromycin or lincomycin, chloramphenicol, tetracycline, and all. So, bacteria developing resistance to a particular drug are resistant to equipotent concentrations of all other types of drugs belonging to the same class. Or it is also possible that the bacteria can show a complete cross resistance or a partial cross resistance. For example, in case of aminoglycosides, you have drugs like gentamicin, kanamycin, neomycin, streptomycin. Now, if a bacteria is found to be resistant to gentamicin, then it is also found to be resistant to kanamycin and neomycin, and then to streptomycin also, but not vice versa. This is partial cross resistance. Complete cross resistance means if a drug is resistant, if a bacteria is resistant to ampicillin, so it is possible that it is resistant to benzyl penicillin also, and vice versa. That is, if the bacteria is resistant to benzyl penicillin, so definitely it will show resistance to ampicillin as well. So Let us see how the resistance is developed. Now, this resistance to drugs by the bacteria are either through the natural mechanisms or through the acquired mechanisms. Now, talking about natural resistance, this type of resistance is genetically bound, and it depends upon absence of certain metabolic processes or pathway within the bacterium. So, clinically, natural resistance is rarely a problem because other drugs are available in the market with antibacterial activity to which the pathogen may be sensitive. So this natural resistance does not pose much of a problem because it is inherited. Now, talking about acquired resistance, this resistance refers to resistance which are developing over a period of time, and this poses a serious clinical problem because once the drug becomes once an effective drug becomes useless for treating infections caused by resistant strains of previously sensitive species. In such a case, a drug which was found to be effective against a particular species of bacteria suddenly becomes useless. So, how does this happen? The development of acquired resistance is basically a stable genetic change which is inherited from generation to generation. Now, there are several mechanisms which are put forth for acquired resistance. One is mutation. Mutation, like for example, um, bacterial resistance develops through mutation. Now, in such a case, a large population of bacteria, of antibiotic susceptible bacteria, is likely to contain some percentage of mutant bacteria that are resistant to the drug. Now, as the drug keeps on acting on such a population of bacteria, which is having a zero point 0.1 or 1% of mutant type of bacteria, this will try to eliminate most of the sensitive members of the species. And what is left is the mutant species now having a scope to rapidly multiply freely and thus creating resistance to the drug. This is what is shown in the diagram to the right. How does antibiotic resistance occur? Second mechanism is adaptation. Now, adaptation is a process wherein the microorganisms at a very low concentration contain a low concentration of certain enzymes which will destroy the antibiotic drug. Like, for example, in case of penicillin, you have an enzyme called as penicillinase or beta lactamase, which is synthesized by the bacteria. And this synthesis is triggered, it is potentiated further in the presence of the antibiotic exposure. So you have enzymes being synthesized by the bacterial cell in order to create resistance. The third type is infectious or multiple drug resistance, which is nothing but the transfer of in vivo or in vitro genetic material coding for the resistance from a resistant bacterium to a sensitive bacterium. This can take place in three different ways. One is transformation, which involves incorporation of a sensitive bacterium of free genes from a drug resistant cell, as shown in the diagram, or it can take place through transduction, where it refers to transfer of the resistant factor, which is also called as R factor, carrying the plasmid by the bacterial virus vector along with its own genes, that is a bacteriophage. Or third, the process of conjugation, which refers to passage of resistant genes, that is the R factors, from cell to cell by direct contact through a sex pilus or bridge. Now, the first case of multiple drug resistance or resistance to multiple drugs was first reported and recognized in Japan in 1959 after the outbreak of bacillary dysentery caused by Shigella flexineri. So you can see the diagram, the process of transformation, transduction, and conjugation as a part of infectious multiple drug resistance. Well, coming to the mechanisms of resistance of antimicrobial drugs, after having seen the basics of bacterial resistance and certain principles behind it, let us see what are the various mechanisms of resistance of antimicrobial drugs. Now, there are many different mechanisms through which microorganisms may develop resistance to antimicrobial therapy. One is increased destruction of the drug. In this case, the microorganisms they produce certain enzymes which destroy the structure of the drug. Second is decreased intracellular drug levels. In this case, the microorganisms will not allow the drug to enter inside the cell. The best example is tetracyclines. They normally accumulate in susceptible bacteria, but not in resistant bacteria. Because in resistant bacteria, uh, these bacteria they produce an inhibitor of tetracycline transport. Next is altered structural target for the drug. uh, Certain targets we have seen where the antibiotics will act, like for example in inhibition of protein synthesis, the 30s and the 50s sites are the most common sites where the antibiotics they act. The structure of 30s and the 40s proteins are so modified by the bacteria to produce resistance, and the drug is unable to interact at such sites. Next is altered metabolic pathway. This altered metabolic pathway, it will bypass the reaction which is inhibited by the drug. Now, some sulfonamide resistant bacteria will do not require extracellular PABA, but some sulfonamide resistant bacteria will start producing their own PABA, which is a direct sulfonamide antagonist. So they will find out a different pathway to bypass the first normal pathway through which the drug was having its effects. Last is development of an altered enzyme. Now, this altered enzyme, the enzyme which is produced, will have a different effect on the metabolic function, and it will be less affected. It will be less affected by the drug. So, in sulfonamide susceptible bacteria, the tetrahydropteroic acid synthetase has a much higher affinity for sulfonamide than for PABA. Whereas in sulfonamide resistant mutants, it is just the opposite. Talking about prevention of resistance, now what happens is the unnecessary exposure to antimicrobial drugs tends to favor the development of resistance. Now, this should be avoided. Normally, antibiotics are taken for treating colds or for small infections which are caused by viruses. Instead, they should be reserved for treating patients whose illnesses have been proved to be caused by some specific microorganisms. So, the steps which will be followed for prevention of resistance or which can assist in the prevention of resistance is first is identifying the pathogen which causes the disease. Second is performing some bacterial sensitivity and resistant testing to be done. And third is selection of proper drug and its drug combination for rational use of the drug. Thank you.